add you, that is translational health research. So this translational health research is very important topic for bench to bedside diagnosis. So this is the actually the meaning of the translational health research, that is the bench to bedside. So you have to basically you have to understand what is the meaning of bench, what is the meaning of bedside diagnosis. So the bench means what? Sorry. Here. So bench means whatever you are doing in the laboratory test. That is called the bed, right? So that is the bedside diagnosis. Sorry, the laboratory is the bench side, and bedside means near the patient. Bench means bedside means near the patient, and bench means the laboratory. So this bench to bedside diagnosis actually we are following in the case of translational health research. So translational research incorporates laboratory breakthrough into the patient care. Whatever we are doing in our laboratory, see, the outcome obtained in the laboratory actually want to apply them. That I told you last time that I like very much these lines, that breakthrough discoveries cannot change the world if they do not leave the lab. So our final goal, whatever we are doing in the lab, so it should be useful for the common people. It should be useful for the patient. So not only you can store your idea in the lab, so bring it to the market, bring it to the industry, right? So this is the actually our principle we are following here, the translational health research. So it is nothing. We're translating our research to the bedside, to the clinical side, right? So you can see here, translational research actually some incorporate some basic science laboratory testing. Then we are going for some clinical research and clinical medicine. Actually, translational research is the not unidirectional; it is the bidirectional, not only forward and backward. So I mean, what sometime we are developing some idea, we are developing some sensor, maybe some device, or we are developing some drug, some vaccine, and we are trying to apply those medicine to the for the patient care. But sometimes we may face some problem, we may face some difficulties, maybe there's some lots of you can say side effect. So we need to bring them back again to the lab, and we need to solve those problems. We should address those issues. We can get some solution for those problems, and we can bring again to the patient care. So translation research is kind of not the unidirectional, not like this lab and then bedside. So we can go from lab to bedside application, then again bedside to bench. I mean laboratory checkup again if we face any issue, right? So this is kind of the translation research. That's why it takes long time. It is not easy method. Like today I develop something and tomorrow I can commercialize those plants or those drugs or those vaccines. Not like this. So translation actually will teach you how we can bring those discoveries for the patient care. So basically, translation research actually expedite the discovery, whatever you're doing in the lab, by actually we are applying multidisciplinary approach, because only one discipline cannot help you from bench to bedside application. It is multidisciplinary. As I said, our course also very much interdisciplinary, multidisciplinary, because we are highly dependent on our healthcare system, highly depend on our basic scientists like biologist, chemist, then mechanical engineer, the chemical, electrical, I mean everybody, everyone can help in this regard. So it is a highly collaborative approach. This research, the translational health research, is a highly collaborative approach. So these things actually stand on the translation research, that is the translation research and implementation science, and this is highly collaborative, and we can innovate some new things for the patient healthcare. So it is very interesting and very important for this course. That's why I'm adding this key point, the translation research. Okay. Students, is it clear what is translational health research?
Yes, sir. Bench to bedside.
Right. It is bench to bedside. Bench means laboratory and bed means near the patient. Okay. Then translational research helps this one. Okay, I will show you some basic idea and some details. And not only basic, some details, details things I will show you now in the next slide. That what's the meaning of translation? Because in the translation, as I said, there is so many component, right? One component you can do some research in the laboratory, then you can bring to the near patient. But for this, you have to overcome so many challenges, right? I think you know, right, in the current scenario, how the COVID vaccine is coming. Actually, in the 2019 already we faced these issues. Now we are waiting for vaccine. So why we need so long time? Actually, although we got some vaccine, but it is also very fast. We got very fast actually this vaccine. Generally, it takes long time, almost 10 years for getting some drug or any vaccine for commercialization. It takes long time. Why it takes long time? So why we should face so many challenges? How we can overcome those challenges? That actually transition health research will guide you. Okay, let's. I will show you in this slide a very good, very informative video. So let's enjoy this. And if you okay, so I think I will copy this link. I will paste it here. Let's
Opening a pill bottle and reaching for a medication is easy to take for granted. This simple act.
Is it audible, guys?
Yes, sir. It's audible.
Yes, sir,
Okay.
it is.
Okay, good. So yeah, this video is very informative, and it will tell you the what the translational research mean. There is so many phases actually in the translational research. T1, T2, T3, T4. There are so many phases, and we should know the definition of T1. What is the translational research of first phase? What is second phase? What is third phase? And how you can bring your discovery to the market. Okay, how you will bring your discovery to the market, that actually you will learn here. Let's see.
Opening a pill bottle and reaching for a medication is easy to take for granted. This simple act is made possible by the work of clinical investigators. But how do treatments such as drugs, devices, and diagnostics develop from an experiment on a lab bench to a product that improves patient health? The process of bringing a discovery from bench to bedside is called clinical and translational research. Clinical and translational research can be divided into 4 phases or T domains. Each has its respective characteristics, but many researchers believe that their work overlaps into multiple domains. The phases of the T domains are different than the phases of clinical trials, but they follow a similar path. Scientists spend years in laboratories testing a new treatment using animal models. This is sometimes referred to as T0. If the treatment shows promise, researchers will give the drug to humans for the first time. This step is the first phase of clinical and translational research, called T1 research.
See, this is the actually translational research. As I said, there are so many pages: T1, T2, T3, T4. So T1 actually your bedside application, bedside your discovery, right? So whatever, I mean, you can apply your knowledge to mice or some some animal study you can do, right, in your bedside your laboratory discoveries, right? So that is just some animal model. So this is fully applicable for not only drug, for vaccines, some device, all of them. So this is applicable for all the discoveries that is related to biological application. Okay, let's.
Is the first phase of clinical and translational research, called T1 research. In this phase, researchers seek to evaluate the safety profile of the drug. To do this, they give the drug to a small group of healthy volunteers to see how the body reacts to it. This research is often conducted through phase 1 and some phase 2 clinical trials. To see if the drug is effective, T2 researchers give the new treatment to patients who have the disease researchers are interested in. T2 studies are bigger than T1, with hundreds of people at multiple medical centers receiving the new experimental treatment. These larger studies make up phase 2 and 3 clinical trials.
See, slowly, so we are going to apply this dose, from small group to the large group. Like this way, we want to see the effect of your treatment, if there is any side effect there or not. So maybe small group, they are not facing any issues, right? But when you apply a large group, maybe they can face, there's so many, you can increase the community, like different countries, people you can collect, and you can apply your discovery, your drug or something, your vaccine there, and you want to see is there any side effect or not, right? So like this way, you have to investigate your discovery is okay or not. Okay.
The results from these studies help to establish guidelines for how to use the drug in the future. If the new treatment is effective, it receives FDA approval. Doctors can now prescribe the drug in practice, and we have moved into the T3 domain. T3 researchers use the guidelines developed in T2 to teach doctors how to administer the drug in hospitals and medical centers. At this point, the new drug is used on thousands of people. Because other drugs may treat the same disease, T3 researchers compare the cost and effectiveness of the new treatment to treatments that already exist in routine practice. Researchers also start phase 4 clinical trials to continue to watch patients who take the drug to make sure there aren't any negative side effects. The new drug is safe, effective, has been approved, and is being widely prescribed. In the T4 domain, scientists study how the new treatment affects global public health. Researchers continue the phase 4 clinical trials that they started in T3 and study thousands of people all over the world to see how the drug is working. T4 researchers often observe whole communities to try to figure out what may be causing the disease in the first place. What researchers learn in T4 studies can often even change public policy. The pill has finally made it into mainstream treatment, but the path to get here was not easy. The process of developing a new intervention takes an average of 10 to 15 years and costs at least $1 billion US. It's hard to believe that only a small fraction of interventions make it through all 4 domains. And most new treatments don't follow the process smoothly in one direction. Often discoveries made in one domain may necessitate that researchers backtrack into earlier phases. Although the road is long, clinical and translational research can have enormous impact on human health.
Okay, so students, it is very interesting, right? Is it very useful topic? Okay, is it fine for all of you?
Yes, sir.
Yes, sir.
Okay,
Sir,
good.
sir, I have a question, sir.
Yeah, tell me if you have any question, please ask.
Sir, the video in the video, the man said that it takes 10 to 15 years to develop, a practical solution to a disease, which can be applied to the masses. So, how COVID vaccine has been just developed in 1 year?
Yeah, yeah.
Does it skipped some of the steps or what?
Yeah, because we are living in that scenario that we need very urgently. So you have seen there is a many the contradictions, right? You saw many in the articles. If you see many news there is lots of contradictions. Is it useful or not? So many people, they are hesitated to take this vaccine. That's why. So because still we need to run we need this trial. Not only the small community, the large community case. We should, we should apply this vaccine. We should know the result, if there is side effect still there or not. But because we need urgently this vaccine in this current scenario, actually we just tested this vaccine only for the small community of the people. But it is not good, although. But we need this vaccine urgently. That's why this is came actually market. But hope this will be useful. But actually, for a effective drug or effective vaccine, effective device, we have to wait for a long time. This is the answer for your questions. Understand?
Sir.
Yes, sir.
Hello.
Tell me.
Sir, actually, you were talking about the effective drug. So this vaccine is not an effective drug, or what you want to say? I don't understand.
But effective means, see, right now, maybe, see, as I said, maybe there some people, they can face some problem, right? You don't know also. Because in this current scenario, some people, they are reporting they have the lots of allergies or something like this, but still we should have the all investigation result, right? That I am going to say, but that actually we don't have for this COVID-19 vaccine, right? So this is kind of for example I'm saying, but still there's so many drugs there, still so many devices there, still many innovation we can do, but we have to go through all these T1, T2, T3, T4, all the stages, and we have to wait for some time. I mean, at least a few years, so that we can overcome all the challenges.
So,
This is
but the vaccine or any drug is actually going through all the four phases: T1, T2, T3, T4. Then how about we came to know about these allergies which people are reporting? Yesterday also 51 cases were reported for the
yeah.
vaccine.
That's what I'm saying. As I mentioned in this video, they said that large commitment, not only the one region, all over the world, all over everywhere, you have to apply this one. Because some cases maybe this virus is not working in some area, right? So then you have to change it, you have to modify it, right? That is the transfer research. You have to go back to the laboratory and you have to modify your innovation. You have to change it, right? So that's the concept. This is the not unidirectional, but that I think. That's not done yet properly. That should be done, I think so, but there is lots of contradiction still there. You can see that in the news, lots of contradiction still there. Some people are not taking also the vaccine, right? This one.
Yes, sir. Yes, sir. Yes, sir. Exactly.
Okay, uh, let's go to the next topic.
Yeah, I think it's almost like malaria, which we used to know that malaria was cured by quinine, and then after many years, also new strains developed, and finally that new drug came, artemisinin, and still people are doing research on malaria that what could be the effective drug. So there are, I think, no penultimate solution to anything. It will keep on
Definitely.
developing, and new strains will come, and everything will work in that direction, I think.
Yeah. That's why our translator teaches us this one. See, nothing we see. As I said,
Yes, sir.
have to apply this one all over the world. I mean, some corner of the world people, they are facing some strain of this bacterium or virus that is that you don't know. Maybe you did not think about this when you develop this drug. But when you try, then you face these challenges. Then you have to come back again to the laboratory. 100% solution is not possible always. So maybe you can say it is 90% effective or say 85% effective, because definitely we will face some difficulties. But you have to come back your laboratory again, and you have to face these challenges. This is so common. Okay, clear, right? It is very interesting, and this kind of research actually we are doing currently, right? And this is very modern and very important topic. Okay, okay, clear. Let's go to the next topic now. Okay, guys, hello, can you hear
Yes,
me?
sir. Yes.
Yes, sir.
Okay, nice. So uh, see here. So translational research means in this case we can use some technology. Let's show you some technology. One technology is nanotechnology. So nanotechnology we can use for different innovations. Like why it is so important, is so innovative, because we can integrate these approaches for different kind of, not only cancer treatment, so many drug. I mean, you can you can apply your drug to a proper area, proper treatment you can use by using nanotechnology. Also, because high throughput, highly sensitive assay of the cancer and any other biomarker, or you can deliver your drug to the you can optimize the drug load. Everything you can do by using the nanotechnology. So it's kind of nano machine you can develop, so that you can send your drug to an actual place, an actual proper place and cell area, so that they can destroy the cancer cells, something like this. So nanotechnology is very innovative, very important for translational research. You can apply your discovery, your your drug you can apply in the actual area, and you can optimize the condition, and you can check how effective your drug or not. So nanotechnology or this kind of the nano machine, this kind of thing actually quite useful for recombinant protein vaccinations, for disease treatment. This is so important. And last thing actually for this translational research, I will show you another topic. So finally our goal after T1, T2, T3, T4, all the stages, so finally we are coming to this point, commercializations, right? So as I think I already told you that we can easily commercialize the device. Let's come back to the biosensor device. So if you want to develop some biosensor device, also you have to face all the stages, and we can bring this device by help of our sensor technology. So I will teach you different kind of lab on a chip. LOC means lab on a chip, so that we can integrate our sensor, and we can use the printed circuit board that I already taught you last class. That we can use the printed circuit board technology. We can use some signal amplification using some chemistry, and we can use some microfluidics for delivery of the sample from reaction site to the biosensor area, and we can measure the signal, right? So everything can be done. And if we can combine this all the sensor with our smartphone, that will be very useful, because everyone nowadays we have the smartphone and we can use. So I will always encourage you to develop something like this, that I already, this is actually one practical application that I did in my lab. So this is kind of very simple, like SIM card, like it's very small chip, very small chip we can develop, and just drop the sample, and we can detect the disease marker within a few minutes. So this kind of chip is highly I can, I'm encouraging my students something like that, to develop something like very simple device, very simple sensor, so that you can commercialize them. And for commercialization of the device also, we have to keep in mind some key points. So how we can commercialize the device. Actually, the next slides here, I'm going to show you some key points. Means for the commercialization of the device, I mean, we have to follow some innovation. I mean, you know, you develop some idea, right? And someone, when some people, they can steal your, they can borrow your knowledge, they can bring your device maybe somehow in the market. So your knowledge or your discourse should be highly protected, and that is called intellectual property. You have to follow all the rule and regulation, you have to file patent. So these kind of things you should know. What is the intellectual property, how you can reserve your, how you can store, how you can you can protect your idea for the clinical applications. So there is so many steps there, but I will give you a brief idea for when you develop some. In this stage, when you start for your startup, or you want to think about some idea, maybe you can come to some our lab, and you can work on some idea, right? When if you get some good result, okay, you can say one day, sir, I got a good result. Now I want to commercialize this. So how we can commercialize this idea, right? You should know this technique, and this all the knowledge you should have, right, guys? So it is very interesting, right? You should know this one. What do you think?
Yes, sir.
Huh? So
We should
this
know.
is so this is very important things. Okay, so let's give you a brief idea. So means you should follow this all the steps when you discover some idea in your lab. So let's see some interesting video. I'm going to show you. Let's enjoy this one. Then I will teach you again.
Fast Forward Medical Innovation at the University of Michigan strives to help biomedical researchers accelerate innovation from idea to impact. It takes commercialization education, translational research funding, and business development support. A simplified version of the product development pathway includes discovery and ideation, invention and prototyping, preclinical testing, clinical testing, regulatory decision, product launch, and post-market monitoring.
See this point, when you want to develop some sensor device, so you should know this all the steps. This is very very important things, so you should keep in mind. Maybe it is not useful for your exam, so this point may be not useful for your exam, but you should know these steps. Maybe I will not give you the questions from these slides, but you should know these steps. It is very very important if you want to launch your any idea to the market. So discover innovation, then you have to try, develop a prototype, then you should go for clinical testing, preclinical testing, then you have to follow the regulatory decision. You should get the approval like FDA. Something like you have to get the approval. If some authority there in India or in the internationally or nationally, you should get the approval from them. Then only you can launch your product. Then you can go for the marketing. Okay.
Although this appears to be a linear pathway, it is often circular and confusing because of other non-technical considerations, including intellectual property, regulatory assessment, and reimbursement and monetization. Let's build a product development roadmap from concept development through product launch for a medical device. The time from concept to FDA clearance to market for a typical Class II medical device can take many years and can cost millions of dollars, so the roadmap needs to be solid before you begin. Here's the plan. Make sure it's needed and that no one else is already doing it. Build a prototype and test it to make sure it works. Find someone to buy it or pay for it. You have an idea that was inspired from a problem or unmet need. Now, it's important to clearly define what that problem is, what's causing it, and what happens if it's not resolved. Next, figure out who else is trying to solve the problem. What is the current standard of care? Are there past failed solutions? The best way to find out if your idea is viable is to ask people, or customer discovery. Test what you think the problem is and if your idea will solve it by asking potential customers.
Yeah, so like this way you have to find out some solution. So if you think about some set up your own startup company or something like this, you have to think about which area you want to explore. So if your idea is really important in this current scenario, see right now if you focus on development a COVID-19 biosensor device, I can say definitely if you get a good solution, easily you can commercialize, right guys? Am I true? Right?
Yes, sir.
So this is the point. This is the point. You have to understand what people need. So if they want this device really, if it is really important, then go for this innovation. So something like this you have to think about. So what is the novelty? Is really useful for people or not? That you have to understand.
Test what you think the problem is and if your idea will solve it by asking potential customers. Your customers are clinicians, patients, health insurance providers, regulatory bodies, hospital administration, etc. What is the value that your product adds to your customer? Remember not to sell your idea, but to ask open-ended questions to find out if the problem that you think is a problem is actually a problem that is significant enough for your customer to change what they are currently doing. From the beginning, make sure that you are developing a diagnostic or device technology that has clinical utility and adds value. Let's assume your customer discovery process was positive and validates your hypothesis that your proposed medical device or diagnostic creates value for your stakeholders. Now, let's prototype. It's important to think ahead of considerations. Be mindful that not all technology developed in the academic setting can be made easily using mass manufacturing techniques. Using FDA-approved materials and/or FDA-approved processes will simplify how your product gets regulated in terms of quality control, safety, efficacy, etc. Incorporate continued customer discovery to assess form factors or how your user interacts with the device, its features, and functionalities. Once you have made some progress on your prototype, start to think about your intellectual property or IP. IP protection is usually accomplished through patents. The design and function of your technology must be useful, novel, and non-obvious to be patentable. Additionally, be careful not to publicly disclose your technology before it is protected, which means detailing your technology in a public setting, such as a presentation or poster, a PhD.
Yeah. So when if you have any idea, good idea, if you get some good result, and definitely we are going for that device development, these things, right? So for that we have to protect your idea, right? For filing a patent. So for that, you should not disclose your idea anywhere. So you can go for some. You can ask some people some questions, like if this. You are going for some solution. You can discuss them for that. You should not discuss with anyone about your technique, how you are going to solve. So that you should not discuss anyone. You should not present your idea anywhere. You should not present a poster or dissertation anywhere. You should not present. So these things, that is the intellectual property you should keep in mind when you are going for any device development or any in discourage for commercialization. So first things, you have to protect your idea.
PhD dissertation or other publications. If you are part of an academic institution or health system, the best way to get IP protection is through your institution's technology transfer office. It's important to contact tech transfer early and often throughout your design and development process. Initiate the conversation by filing an invention disclosure as the first record of invention. Your tech transfer office will work with you to determine a meaningful and useful method of technology protection. At this point, you have a prototype and have talked to your tech transfer office about IP. Now it's time to decide how to test it. The FDA will define the testing plan. The overall timeline and cost to market is varied depending on the complexity of the device and risk to the patient. The FDA allows marketing of devices that are safe and effective and separates medical devices into 3 classes. Class 1 devices pose the least risk to the patient and frequently only require a registration as manufacturer of record with the FDA. Think elastic bandages or dental floss. Class 2 devices pose a moderate amount of risk and most often require a pre-market clearance from the FDA before they are sold. Class 2 devices are the most common and include things like pregnancy tests, infusion pumps, and monitoring devices. Class 3 devices pose the most risk to patients, are generally life-sustaining or implantable, and are subject to the most rigor by the FDA, requiring clinical trials for approval. Think implantable pacemakers and breast implants. As you move from Class 1 to Class 3, the time and cost from initial development to market increases because the amount of evidence required to demonstrate safety and efficacy increases. Let's focus on Class 2 devices, the most common, with a typical 510K FDA clearance process. The foundations are intended.
Yeah. So this is the things I will share this video. So the things I already discussed with you, the important points, so you can understand so how we are commercializing the device. Okay. So this is the introductory classes for the all our biosensor biodevices. I already taught you. So now I will start some new topic today. Okay, students. So I will ask you. So, do you have any questions up to this slides? So, if you want, I can discuss with you. Otherwise, I can go forward to the next topic. So, please ask me if you have any questions. Please ask me. So, do you want any clarification anywhere? Any point? I want to discuss with you again. Or if it is fine, then I can go next topic. What do you feel?
Hello, sir.
Ah,
Hello,
tell
sir.
me.
This is Aman.
Ah,
So,
man.
sir, I just little curiosity, sir. I wanted to ask, sir, if you create a particular medicine, like you were asked telling us. So, sir, how did you manage to see the long-term symptoms? Like, if you're injecting in person's body, so how
Yeah.
did you see a long-term vision that it will be beneficial or it will not have a any side effect? How did you check it? Check it in that was my
See,
curiosity. That just like. of COVID
Huh.
vaccine we are using?
Yeah, yeah. See, I taught you last time. So if you want to see the long time effects, then we are doing the continuous monitoring, right? We are monitoring continuously. So we are developing some device. I mean, as for example, for this course, I'm telling you, for this course, generally we are developing some continuous measurement, some device like this kind of thing, some flexible biosensor we can attach, right? So and it will send wirelessly all the data to your portable device, and you will get all the values. Maybe you can use it for a whole day, then maybe for a few days you will get all the values, and you can predict the effect of this vaccine, or effect of this drug, or effect of the or anything. You can I mean, you will get some idea, then you can conclude finally. So something like this way, you can monitor continuously by developing a device. It is very, very important things for continuous measurement, right? Okay.
Thank you, sir. Yes, sir.
Huh. Yeah. So in this subject, that's a very important, because you will get so many ideas, some different topics, not only one area, but it is interdisciplinary. All the idea you will get it. I mean, everyone like chemist, physicist. I mean, the mechanical guys, I mean, the electrical guys. You all will be involved equally. Okay. So, any more questions here? Okay, so good, very good. So I think you understand this all the topics I taught up to this to these slides. Okay, now comes to the next point for this course, that is nanomaterials. Now I am going to teach you how we'll develop some sensor device. Now uh, we are entering slowly we are entering to the biosensor device development. So until now, I just show you all the scenario. This is the magic things for this biosensor. They can do this thing, they can do that thing. But how, right? So this question should have in your mind, right? How we can do this? What is it, how we can solve this problem? Maybe I can teach you so many solutions. I mean, this is some generic. As I'm saying from the first day, I will teach you not the specific sensor, some generic sensor. It can be useful for all kind of the disease. Hmm. So you can use this knowledge in your future also, for your master project, for your dissertation, also for your future research. You can think. Okay, so let's come some nanomaterials. How we can use the nanomaterial for device development. So first of all, I want to show you that what's the nanomaterial. So material with at least their size or dimensions should be within 1 to 100 nanometer, very small. Uh, so due to their small size, so nanomaterial means the size should be very small, within 100 nanometer, and due to the small size, most of the they are showing some unique behavior. Nanomaterial, they are showing very unique behavior. There's due to the, I mean, you can, the effective surface area will be different, surface energy will be different, and then the bulk material. So if you think about gold nanoparticles, so their activity, their surface area should be fully different than the bulk material, and it will it can act as a catalyst, and it will uh, react with some chemicals, and it will enhance the sensing activity. Signal to background ratio will be increased. So I mentioned many times what signal and background. So signal means what you are expecting from the biosensor. Background means, so without adding any sample. So when you are not adding any sample, but still you will get a somehow some output, that is in the form of current or in the form of charge, that is background. So without adding any biomarker or any solution, or I can say maybe you are going to measure some cancer biomarker, right? But your sample maybe does not contain any cancer, but still you will get some value in the form of current or in the form of charge from your device. That is the background, but not, or you can say noise. But if your sample contains some cancer or any other protein that we are going to detect, so in that case, whatever you are getting, the current or charge, that is the signal output. That is the your signal. So signal to background means if your background is very low, your sensitivity will be very high, right? For your biosensor device, if background low, you will get the higher sensitivity. Actually, nanomaterial will help you to get a higher signal to background ratio. Hmm. So I will teach you again, say what is the signal, what is the background by using a sensor device. I will show you later again. Here in this slide, I will show you some nanomaterial by different shape, different size. Actually, by changing the nanomaterial shape and size, we can change their activity. Because the surface energy, the surface area, everything that can be changed. Also, we can synthesize some core cell type. I mean, outside one material, inside another material. So because of the different surface energy, they can behave differently. So we can use some spherical, some nanorod, some prism type. Because of the different shape, their activity also will be different. So this is kind of the investigation people are doing. People already did many investigation by changing their shape, by changing their size. So this is also something. I mean, research still going on. How nanomaterial behave if we change their size, change their shape, change their composition. Like maybe we can something like this. I can say uh, just wait. I mean, if we like Fe nanoparticles, like iron. Fe means iron nanoparticles. But maybe gold and Fe, so both you can mix together, then you can form your nanoparticles. So these two mixture, then this is kind of the hybrid nanoparticles you can form. So this hybrid nanoparticle may behave some different way. Maybe in this case, you can get higher signal to background ratio than only the iron nanoparticles. So something like this you can investigate in the lab and you can find their behavior in the biosensor. So here just I have I just put there the different size of the materials and then their size. I mean, where the antibodies, virus, bacteria, protein, different kind of chemicals. What is their size and which range the I mean in the nanoparticle size. I mean, which range there? There's actual. I can say that if this material size is 10 to 15 nanometer, maybe they can behave something different. If their size 50 to 100 nanometer, maybe they can behave something different, right? So these things actually we are investigating. Okay, next come to the another things. So I'm saying always the nanomaterials, but there is different kind of I'm actually classifying now different kind of nanomaterials. You can say some metal based nanomaterial, metal based, and another one you can say non-metal based and non-metal based. So metal based nanomaterials like you can say gold nanomaterial, platinum nanoparticles, and silver nanoparticles, and iron nanoparticles. Some different metallic nanomaterial you can form and you can use them for biosensor development. Some non-metal based like some carbon based, I think you know already, like CNT, that is called carbon nanotube, or graphene. Hmm. Or you can say carbon nanorod. Here you can see some shape. So you can see some uh, A is the fullerene, some like football like shape, fullerene. This is a sheet like the graphene sheet. This is the nanomaterial also. Hmm. So some carbon nanotube, some carbon nano onion, nano diamond. So different kind of diamond shape if you can develop. So different shape if you can develop, their activity also will be different. Hmm. So like this way, you can use them for sensor development. So here actually I'm going to show you a actually all the steps. Maybe I will teach you not only in this slide, maybe after the maybe today's class, I will bring some picture, like some step by step how we can develop a biosensor, right? Step by step. That I will teach you slowly. Just today, I want to show you a whole picture of the biosensor. See, in the left hand side, you can see a picture that is actually ACM image. I will tell you what is the ACM. That in this microscope, actually we can see very small here. You can see the gold nanoparticle. ACM, the scanning electron microscope. In that microscope, we can see very small, almost it is 10 to 50 nanometer, 50 nanometer gold nanoparticles. We can see it is something like this. So we can fabricate, I mean, some sensor surface. This is called the sensor surface or transducer. I taught you, right? What is the transducer? Transducer is the surface where we are immobilizing all the materials like antibody and other nanoparticles. That is the transducer. So here, GC is the glassy carbon electrode where we will immobilize the maybe gold nanoparticles. Why? Because of the nanomaterial size, it can increase the surface area. It can increase the surface energy. Because of this, we will get higher signal to background ratio. We will get higher signal to background ratio. So we first will immobilize some gold nanoparticle on the surface. Then on that surface, we can immobilize our antibody. So instead of gold nanoparticle, only we can immobilize the antibody also. But if we immobilize the nanoparticle, maybe we will get a higher surface area. Lots of antibody we can immobilize on the surface. So before nanoparticle, maybe we can, for example, just think, maybe 50 number of antibody we can immobilize on the surface. But if we immobilize many, I mean, if we can drop some gold nanoparticle, so we can increase the surface area. In that case, we can immobilize almost 5,000 antibody on the surface. So huge number of biomaterial we can immobilize by the help of some nanomaterials. This is the utility of the nanoparticles on the sensor surface. So we can get higher signal to background ratio. So this is so many steps actually here in this paper shown. I mean, we can immobilize some nanomaterial, then go for some chemical treatment, something like this. That actually I will teach you later. Just I'm just showing you here step by step. Actually, people can form the biosensor surface like this way. So don't worry, I will teach you in the next slides also how we can form the biosensor surface, something like this. uh, See here. So this is the full biosensor. So this is the full biosensor. So what is the biosensor? I think I already taught you the definition of the biosensor. Can anyone please tell me the definition of the biosensor? Anyone? Hello?
Hello, sir.
Yeah.
Hello. Sir,
Hello.
can you hear
Yeah.
me, sir?
Yeah, I can
Yeah.
hear you. Can you tell me what is the
Yeah.
definition of biosensor?
Yes, sir. Biosensor is a device which uses biological materials or to detect whatever we want to detect, and sometime it it detects using metals also, or any non-metals to detect biological materials. It is called biosensor.
Yeah, so it can detect the biological material, but it should be very specific. So it is kind of a tool. So biosensor, it is kind of a tool that can detect some specific biomarker. You can say specific. So specific word you have to use. You know why?
Yes.
Maybe you are developing some biosensor. If it can detect for cancer, right? So it should be very, very specific for cancer only, not for malaria, not for dengue, right? So it is very important point. It should be very, very specific. Okay.
Okay, sir.
Otherwise it's good. Okay. So like this way. So this is the, you can see here I'm going to show you one picture. That is the biosensor. So what is the target here? This one. So this one is the target. This marker we are going to detect. So this target can be PSA or mouse IgG. So this is, we are saying, the target. So PSA is called prostate specific antigen. This is kind of the cancer marker for that cause the prostate cancer that we can detect. So mouse IgG, just leave it the mouse IgG. Just think about only one. That's only one, the prostate specific antigen, PSA. So we are going to develop a biosensor for prostate cancer. So what we will do? So first we have to choose a transducer, right? On that transducer, we will develop step by step our biomaterials. Then we will detect our prostate cancer. So here ITO. What is the full form of ITO? ITO is the indium tin oxide. So this is kind of nanoparticle. It's the very, very small indium tin nanoparticles actually coated on the glass surface. Hmm. So on the glass surface, indium tin nanoparticle already coated. So as I said, if glass surface coated very, very small nanomaterial, then readily we can increase the surface area, right? Huh? Okay, students. So
Yes,
is
sir.
it clear why we are using the nanomaterial? So
Yes, sir.
if we use the nanoparticle on the glass surface, we can easily increase the surface area. So then what we are doing? So some linker. This is kind of some linker. Some linker, some protein. So I will teach you what kind of linker generally we are using later. I will teach you just for today's preliminary class. So I'm saying some linker we can drop on the nanoparticle surface. Some linker. On that linker, we can add some antibody. So antibody can be linked on the sensor surface by the help of linker. So this is the sensor is ready for the biomarker detection, for your target detection. So this is actually your biosensor. So this is the sensor surface is ready for detection. So what you have to do? First you have to choose a transducer, that is the sensor surface. On that sensor you have to drop some linker. By the help of linker you can add some antibody. Antibody is the bioreceptor, right? I taught you already what is the bioreceptor. So bioreceptor can bind with your target, right? What is your target? That is some biomarker like cancer. So in this case PSA is your target, right? We have posted specific antigen. So this is your target, so that you are going to detect. So that specific antibody, so if you are going to detect some cancer antigen or some target, you have to immobilize on the surface that specific antibody. Clear, guys? So you just ask me. If you're not clear, I will repeat again. Is it clear or I should repeat?
Sir, I have one doubt, sir.
Yeah, tell me.
Sir, you said linker is connected to this nanomaterial ITO.
Huh.
Sir,
Yeah.
obviously, yeah,
Huh.
you said the linker is also a protein, some kind of protein.
Huh.
Yeah,
It can be or cannot be, but you can use some kind of protein. Yeah.
yes, sir. I'm assuming it is a protein, and if protein is there, protein is obviously bigger than this nanomaterial. Why you're using nanomaterials down? If it is already bigger than this,
Huh.
uh,
See,
yeah,
okay. See, this is also a good thing so you can think about. So this is a surface. So there is lots of nanoparticle. So you are adding some linker, so that can be adsorbed, not only one nanoparticle. Maybe they can adsorb on the two or three, maybe whole area in like this way. The linker can be like this. So if you not use nanoparticles, uh, just think about. I can show you some view like this way. If this surface is there, nanoparticle on your surface, then maybe your surface will be like this, like spherical style, not like plane. So because of that, so you can get the more linker on the surface. So more linker means more antibody will be on the surface. If it is flat, so maybe only maybe two linker will be there, and you will get only two antibody on the surface. But because of the spherical size, you will get more linker on the surface and more antibody on the surface. And more antibody means more target can bind on the surface, and more target means you can detect your biomarker from some maybe in the long range, like picogram per ml to maybe microgram per ml. In the long range, you can detect your target. Clear?
Yes, sir.
This is the utility of the nanoparticle.
So I
So,
have a question.
tell me.
So can you repeat the term transducer?
Okay. So I think
ITO, ITO with the sir, ITO on ITO we immobilize the nanoparticles. Then we added the linkers. So this whole term can be called as transducer.
no. See, transducer. I think did you miss the last class, right? The transducer when I taught. I think,
Yes,
right?
sir. Yes, sir.
Yeah. So transduction, nothing. It is just like a surface, where we are immobilizing our all the materials and we'll apply some input like some potential, and we'll get some output in the form of something like current. Hmm. When we'll integrate this one with some mobile device, so then we'll get some output like current. So what is the transducer? Transducer is something like a surface where we are working on. Huh. So we are immobilizing the materials, we are applying some potential, we are getting the output by the help of this solid surface. Huh. So this solid surface is called transducer. Okay, clear? So on that
Clear.
surface, maybe we can immobilize nanoparticle. On that surface, we can immobilize linker, antibody, everything. Maybe where you are working, sensor. That is the transduction. Okay. So on that, so here we choose actually as for example ITO. ITO means indium tin oxide nanoparticle coated glass surface. So this is your transducer here. Just so I will give you so many transducer example. Then I will teach you which one is the best, which one not. So how we will select the best one. Hmm. So now few classes, this continuously few classes I will show you the many transduction name. So today I am showing you one transducer, that is ITO. So ITO is nothing but just indium oxide glass surface where we are immobilizing the selective antibody by the help of some linker. So I will teach you again. So what kind of linker you can add? So what is the name of the linker? So what is their characteristics? That also I will teach you. But just in this class, I'm going to show you how biosensor we can develop. Okay. So at first you have to choose the transducer, then you have to add some linker, then antibody. So you can add not only antibody, any bioreceptor, but just for example, I will teach you, bioreceptor, just antibody. I will stick on this example. Otherwise, you'll be confused. So antibody is your bioreceptor. So if you want to detect the prostate-specific antigen, we will immobilize specific antibody for prostate-specific antigen. Okay. So now you will drop the sample. So this is the sample you are going to drop on your surface. Which surface? Your sensor surface. So if the sensor surface contain your target, what is your target here? PSA, right? Guys, so what is your target here? PSA, postage antigen is your target. So if your sample contain PSA, then it will bind on the sensor surface, because they are very specific for this antibody. So this antibody will bind with this antigen. So what is the antigen? Antigen is your protein marker that you are going to detect, that marker. That is your biomarker, or that is your target that will bind with the antibody. Clear, students? Or I want to repeat again. Is it fine? Everything okay?
Yes, sir.
Sir,
Yes,
that
sir.
that AU part is not clear. What is AU above the
No, yeah, I didn't say yet. I will come. I will come.
okay,
Wait.
sir, okay.
Okay. So these things I will drop. Right. Then your target. See here. So here target already on your surface now. Post-specific antigen. So it is surface. Now, as I said, we need some signal amplification. Amplification. So why we need signal amplification? To get higher signal to background ratio. So when you are immobilizing some linker, some protein or antibody on the surface, so you will get some background current. So when you apply some input, so you are applying some potential like input, then you will get some in the form of some output like current or charge in your device. So because of this kind of material there, and we will use some reagent, some chemical. So here you can see NaBH4, like sodium borohydride. AP is called aminophenol. So this kind of chemical we will use, or NO2, this one, the NP, this is called nitrophenol. So this kind of chemical we will use in the biosensor surface. Why we will use this kind of chemical? Because they will react each other and they will generate some current, or they will generate some charge. That is your signal. Hmm. So signal means, so after adding your target, so or after adding your analyte solution, when you are applying some input in the form of some potential, something like this, when you are getting the output, but although maybe you are not in your sample may not contain target, but still you will get some current, right? Or still you will get some charge in the form of some output charge or output current. That is your background. And what is the signal? See, when you add your sample, if your sample contain your target like PSA, then you can add some another antibody. Another antibody means that antibody actually it should specific to their target. Uh, it should very specific to that target, so that it will bind only this antigen or this target. It will bind, and that antibody called is the secondary antibody. Okay, so I'm going to write here. See, this antibody is called primary antibody, which is bound already on the transducer surface, and another antibody that is called secondary antibody. So secondary antibody actually, after adding your target, you can add another antibody that is conjugated with AU. AU is the gold nanoparticle, NP. So AU is the gold nanoparticle. So you can bind your gold nanoparticle with your secondary antibody. So this is your AB2. AB2 means secondary antibody. And this is AB1. AB1 means primary antibody. So your primary antibody helping you to bind your target, and secondary antibody actually help you. How it will help? So it will bind after adding your target. It will bind on the your target, so that nanoparticle will come to your sensor surface. So basically, we are incorporating here nanoparticle on the sensor surface by help of secondary antibody. So here, as I said, the secondary antibody already conjugated with the gold nanoparticles. So if your surface contain your target like prostate-specific antigen, then only secondary antibody will bind. Otherwise, your secondary antibody will not bind. If your surface does not contain any target, then secondary antibody will not bind. So if surface contain target, then secondary bind, and secondary antibody contain the gold nanoparticles. So basically, indirectly, you are adding gold nanoparticle on the surface if your sample content target or biomarker like cancer marker. So if the sample content cancer marker, then only gold nanoparticle will bind on the surface. Guys, make it clear. I will otherwise I will repeat you. Just make it clear. Like this way, we are forming the whole sensor on the surface. Is it clear until here?
Sir, I have one
Sir, can
question.
you repeat?
Tell me.
Sir, you told in the end that we have to add this secondary antibody, but how we will know that we have to add or not? Because ultimately our purpose is to just check for the PSA.
Yes.
So then how then you are saying that we will add secondary antibody and we will let it will only bind with the PSA. Then
So
without knowing this, how we will add? My question is this thing.
okay, so see, actually the question is why we are adding secondary antibody. So secondary antibody we need here because so say you are very true. You are going to detect PSA cancer antigen, right? So if your sample content prostate specific antigen, then it will bind on the surface because primary antibody very specific to that prostate cancer, right?
Yes, sir. Yes, sir.
Okay. Now, so your surface content already present, but we are going to detect that prostate antigen with the help of some chemical reaction. Okay, with the help of some chemical reaction. So this is some chemistry I'm going to teach you. So for that reaction, we need some catalyst. We need some, we can say some enzyme. But how? So now the question. So now the question is, so how we can generate how we can incorporate a chemical reaction on the surface. So how we can create a reaction. For that we need some tag. So here gold nanoparticle something like the tag. See, if you have the target, then only secondary antibody will bind, then only gold nanoparticle will bind, otherwise it will not bind. Now, if gold nanoparticle on the surface, then this NP, that is called nitrophenol, it will react with the gold nanoparticle with the help of one chemical that is called sodium borohydride. Sodium borohydride is a reducing agent, reducing chemical, so that NP will transform to the AP. AP is called aminophenol. So it is so actually this will be reduced to the AP by the help of sodium borohydride if if your solution contain gold nanoparticle. So if you do not have gold nanoparticle, then this reaction will not be feasible. This reaction not possible. Now, so if you have done aminophenol, then means AP, then this AP actually will react on the surface. So here actually I use the FC plus. FC is actually iron compound. This is actually compound. Iron compound. It is called ferrocene. FC is the ferrocene. So this ferrocene and AP they will react together. So just think about. So if you have NP, mean nitrophenol, it will not react with the FC, means ferrocene. But if you have AP, aminophenol, then it will react with the FC and it will get high signal. So basically, you want to generate a very good signal to background ratio. So as you have gold nanoparticle, that's why you are generating AP. And AP actually reacting with the FC plus and you are getting very high signal. I mean this chemical reaction actually helping you to generate very high signal. Actually for this reaction we are generating the electron. That electron actually we are measuring in the form of current or in the form of charge. Okay. So if this reaction if there is no reaction, then this electron will not flow. If there is no electron flow, then the current or charge you will get very low. Hmm. So for getting high electron flow or high signal, you need this reaction. And who is helping this reaction? Actually, gold nanoparticle is helping on this reaction. Okay, I will repeat again. Just make it clear. So it is very very important step. You should learn or you should know first the each step. You should uh, clarify all the steps in the beginning few classes. Otherwise, you feel that other classes also not clear for you. Okay, so is it clear or I should repeat? Students, is it clear?
Sir,
Yeah, tell me.
sir, uh, starting from that AU part, sir,
Huh.
our motto is to generate high signal to background ratio. Correct? Am I correct?
Right.
So it means indirectly, I can say that we have to generate an electron so that we can detect that there is cancer biomarker.
Right.
So all this process enables us to know that whether this PSA is there in the annulate or not.
Yeah, pH is there and the not. Right.
Okay, so fine. This was I was asked.
Yeah. See, so if PSA there, then only goal. So what is your name, please? So actually, I should interact with you always. I should understand. So who asked this question?
Sir Bhupendra sir.
Okay, good, Bhupendra. So you, that is very good question. So just I want to make it clear again, and this again I will ask all of you. Please make it clear. So as the Bhupendra asked here, see, mm, wait, here, see, if your surface contain prostate-specific antigen, then only gold nanoparticle will bind. Right, Bhupendra? Is it clear for you?
Yes, sir. Yes, sir.
Huh? If
Yes.
prostate-specific antigen there, then only AU will bind. Otherwise, AU will not bind because the secondary antibody that conjugated with gold, that is very, very specific of the on the target. And if gold there, then only NP will react with NaBH4 sodium borohydride with the help of gold nanoparticle, and it will form the AP. And this AP will react with the ferrocene Fc plus, and it will generate the electron. That electron actually we are measuring. Clear?
Sir, in all this reaction,
Yes.
sir, can we this generation in the side product, is it of no use, or how does it interfere with the reaction?
Yeah. So there is some other interference also effect there. Also there is some. I mean. Not only interference effect, there is some other side effect also there that I will teach you. So just today I am going to show you how biosensor surface can be developed. Okay, just small part I am going to clarify you first. Okay.
Okay, sir. Okay, sir.
Okay. Okay.
So,
Sir,
sir,
I
but
have one doubt, sir.
if
Tell me what is your name?
Sir Dushyant Kumar, sir.
Listen, Kumar. Okay, please ask.
Yeah. Sir, there are primary antibody and secondary antibody, and they both are looking for this antigen. So, will there
Yes.
not be competition between these two, sir? Like before only this secondary antigen covers all this primary PSA. Will the primary antibody will detect that one, sir?
Yeah, that's also very nice question you ask. See, just means I want to make it clarify. I will just clarify you. So first we are immobilizing primary antibody, right? So this is your actual biosensor and it is ready for the your biomarker detection, right? Up to here. Then you are adding your antigen, means your sample. So you are adding sample. You are not adding any secondary antibody. So this primary antibody, that is antibody 1, already on your surface. Huh? So now you are adding the sample, then sample will bind. So one step actually we are following, that is called washing step. Hmm. So after adding your sample, if your sample content target, then it will bind. Then there is lots of unbound. Your target can be there. So when you are washing that unbound one, they will go out from the surface. They will not on the surface, right? So only target will bind, and other if you have the excess amount, they will be removed by the help of washing step. Okay, then we will add the secondary antibody that is conjugated with gold nanoparticle. So there will be no competition.
Okay, sir. I thought the secondary antibody will also be there in the biosensor. So
No, secondary antibody, it is on a part of your biosensor, but it will be added in the after adding your sample and washing step, it will be added. It is also part of the biosensor, but it
okay,
will be
sir.
added in the next step.
Yes, sir.
Okay,
Now it's clear, sir. Thank
that's why there will be no competition. Good question. So please ask something like this questions. I will clarify you. Any more
Sir,
questions?
sir,
Yes. Who are you? What is your
sir,
name?
Vibhuti, sir, sir, Vibhuti.
Bibhuti. Okay.
Sir, what kind of washing buffers could you use, or
Yeah.
will it hinder with the biosensors?
Yeah, yeah. So that see that's I told you. So name of the component, everything I will slowly I will teach you. So that's today actually going to show you just the how we can form the biosensor. Okay. So
Oh,
generally, just for your information, but washing buffer that can be some surfactant we are using in the washing buffer, some BSA, biovincinum aluminum or 2020, something like this. So there is some surfactant generally we are using. So they are helping to remove the unwanted pieces, and they will help to block the surface so that other unwanted protein will not be absorbed on the surface. Just for your information, but I will come that point. Hmm.
okay, sir.
Okay. Akash Sharma. Akash, are you there?
Yes, sir.
Okay. So is it okay for you? Everything clear? This how we can form the sensor?
Yes, sir. Like I was trying to understand, but I will look into the slides also afterwards.
Yes, yes. So yeah, so like this way, we are forming the biosensor one by one sensor step. Uh, so, so in the next class, now I will teach you how this antibody can be bind. Now this antibody how we can immobilize on the sensor surface. How this linker can bind with this antibody, right? So these things still I didn't teach you, right? So how the linker can bind with the antibody, how gold nanoparticle can be conjugated with the secondary antibody. So this linking process, this is the linking process. So the scientific name is called conjugation, the conjugation process. So this conjugation process I will teach you in the next class. So how you can bind this primary antibody on the surface and how we can bind the secondary antibody with the gold nanoparticle, or not only gold nanoparticle. So here the secondary antibody we can bind with any enzyme also, enzyme or gold nanoparticle, not only gold, any kind of nanoparticles. So why we need this tag? This is called tag. So why we need this tag? They are actually helping some reaction like this. So if this tag or this is kind of actually catalyst, enzyme, nanoparticle, they're catalyst, right? They're helping this reaction, this catalytic reaction, they're helping. So how we can conjugate this kind of the catalyst with the secondary antibody? So that I will teach you in the next class. Okay.
Yes.
Yes, sir.
Okay, guys. So I will request you to please join the class as soon as we start. So like today we are starting from 7 and next class we start from 3 p.m. Wednesday. So please join as soon as we start so that you will not miss any topic. Otherwise it will be difficult to understand. Okay. So today's 46 students already joined, but at the beginning I got only 30 something like this, 25. Okay, all. Everything fine?
Okay, sir, fine.
Okay.
Yes, sir.
So
Fine, sir.
So I am taking attendance now. So just I can download your attendance list. Okay. So at the end of the class, I will take the attendance. Okay, done. Okay, so thank you. That's all for today. See you in the next class. Thank you all. Good night.
Thank you, sir.
Thank you, sir.
Good
Good
night.
night.
Thank you, sir.
Hmm.
Thank you, sir.
Thank you. Thank you.
Yes, sir.
Thank you.